Hi, Miss Patterson here. Today we're going to cover lipids, part of the macromolecules unit. So lipids, they're considered hydrophobic. So hydro means water, phobic means fear of. So hydrophobic means that lipids don't like to be around water. They're going to be repelled by water. These are not considered polymers. Remember, a monomer are small little bits and pieces of a macromolecule. I can link them together and make a long chain, which becomes a polymer. Lipids don't do that. So lipids will have the monomer, which will be like a triglyceride, a phospholipid, something to that effect. But I'll have a glycerol and I'll have a fatty acid tail. So they're not assembled from a variety of components. They're just basically two pieces, and that's it. I have the glycerol part, so you can see this part here is the glycerol. This is going to be my fatty acid tail. And what do you notice about these fatty acid tails? You see that they're all carbons and hydrogens. So when we talk molecular wise, carbon and hydrogen are considered covalent bonds, because if I look at electronegativity chart, these are very close in charge. So these are going to share electrons back and forth evenly. One is not going to have more of a charge than others. They're not going to be like oxygen here. Oxygen, remember, very electronegative, very greedy for electrons. It wants all those electrons. So lipids will come together with a certain type of bond. There'll be ester links, ester bonds, and it's a dehydration synthesis. Remember when we talked about carbohydrates, the water got pulled out of each monosaccharide, and that bonded them together to make a polysaccharide. The lipids and all these other macromolecules do the same thing. Water is going to get removed to put them together, so they're going to dehydrate. My glycerol, I have these hydrogens that sit on the end. I have these OHs, these hydroxyls, that sit over here on the other end of my fatty acid tail. They're going to come together and make water. The water is going to leave, and then it's going to pull that oxygen and those carbons together, and that's going to create my bond. So I might have a triglyceride, and it's a tri because I have three tails, and then I'll have three water molecules that will come out of it. We do this with dehydration synthesis. Another one is called condensation reaction, same thing. These are considered hydrocarbons because you can see, what do I have? A bunch of hydrogens and a bunch of carbons. So hydrocarbons are nonpolar because of those tails. I can have something written out like this. You'll see this sometimes, and scientists can be lazy. They want to make a shorthand. When you go back and you look at those hydrocarbons, you see all those carbons and all those hydrogens. We just don't have time to write all that stuff out. So we do a shorthand. So all of these little peaks and valleys represent a carbon. A carbon will be here, a carbon's here, a carbon's here, and we just assume that each of these points represents a carbon. And we know off those carbons I'm going to have hydrogens. So it's assumed if I have a carbon here, I'm going to have two hydrogens coming off of here. If I have a carbon here, I'm going to have two hydrogens coming off there. So I'm going to have that on each of these peaks. I don't have to write that out. Same thing here. Carbons, carbons, hydrogens attached to those. But see this spot right here? This right here is telling me I have a double bond between carbons. So I have a carbon, and I'm going to have a double bond here. Over here you can see I have three double bonds, one here, and one here. So I can show real quick a shorthand of those hydrocarbons. I got two types of triglycerides. I have saturated and unsaturated. This one here is considered saturated because each fatty acid tail is saturated with hydrogens. I have all single bonds between all of my carbons and my hydrogens. This is going to make it nonpolar. This is going to be commonly produced by animals. So animals produce saturated fats. So if you eat meats, if you eat stuff from animals, chicken, beef, bear, deer, okay, you're going to have some type of saturated fat in there. Some will have more than others. These are going to be considered solids at room temperature. So if I have butter, I take fats from a cow, or I make butter from a goat. All that's going to have some type of saturated fat, and that's what's going to help my butter solidify. You also see these three tails, how they're straight. These three tails are going to pack tight together. So you think, all right, I've got these three tails. They're going to pack real tight. They're going to lay on top of each other. This is going to allow the fats to solidify at room temperature. These are going to be bad for cardiovascular health. These like to stick in the arteries and blood vessels, so these can start to build up and cause plaques. Unsaturated fats, you can see the difference. These are all straight. Because remember, my 3 were straight. This one here, I have 2, but this other one's kind of like kinked. It's not going to allow it to come up and push up against these ones here. So this is going to create a space inside my lipids, my unsaturated fats, that isn't going to allow it to be a solid at room temperature. So these are still nonpolar, my hydrocarbons. I'm going to have at least one double bond. I can have more than one double bond in a fatty acid tail. These are going to commonly be produced in plants. That's why a lot of plants are pretty good. You know, you're going to have some that are going to have some type of saturated fat, but it's not going to be a high content. But I can have plants that are going to be unsaturated fat. So vegetable oil is better than like lard. So those type of fats are not as cohesive into your arteries and your blood vessels. These are going to be more of a liquid at room temperature, so olive oil, peanut oil, vegetable oil, those are all going to be some type of liquid at room temperature. Here's another shorthand version of it. I can see here that I have a plant. It's going to be double bonded. I'm going to be an unsaturated. These are going to produce kinks on that fat. So here I've got my 2 pieces. Saturated is straight, unsaturated is bent. So here my saturated, I can see no double bonds. Here unsaturated, I have a double bond, so this is going to prevent the packing of these tails, so this will be more fluid at room temperature. The function of fats, these are going to be some type of energy store. So these cells are going to store certain types of lipids. It's going to have twice as much caloric intake per gram than carbs. So it takes more to burn fats, but fats are more efficient at burning energy, I should say that. But hey, they even used them back in the day, candles. They used whale fat and whale oil in order to burn, and that was a lot better than like burning paper or something. Beeswax, they use beeswax too. That's a type of lipid. This is going to help protect organs. It's going to help insulate. So if you have fat stores, it's going to help keep you warmer. You see this with animals that are in Arctic areas. They have a lot of fat in certain areas, and that helps to insulate them. We have what's called adipose tissue. So the adipose tissue is the type of insulating fat that we have in our bodies. We have brown fat and stuff like that. Phospholipid, we're going to see these in cell membranes. You're going to have a hydrophilic head and a hydrophobic tail. This is going to be water loving. This is going to be water fearing or water hating. So here I can see my saturated fatty acids, all these hydrocarbons. Unsaturated, I'm going to have a little kink right here. This is all hydrocarbons. Up here I have a lot of oxygens. So remember we talked about oxygens, very electronegative, very polar. It's going to start pulling water and stuff into it. So I'm going to have a hydrophilic head, hydrophobic tails, and it's going to line up in a certain way that's going to help make membranes. We call this amphipathic, and this means that you have a hydrophilic side and a hydrophobic side of an object. So here I have a cell membrane. This is what's called a phospholipid bilayer. Up here is my hydrophilic, and then here in the center are all my hydrophobic. So if this was a cell, I'm going to have my aqueous environment outside. I'm going to have an aqueous environment inside. So these hydrophilic heads are going to be very happy. This part here is not. So they're going to naturally turn into each other. And this is going to be a water-free area for the most part. So I'm going to have this bilayer one layer here, one layer here. That's going to help to regulate what goes in and out of a cell. I can have different shapes. I can have a sheet. I can have them where they're all balled up and all the tails are inside. I can have like a bilayer kind of sphere where I can have stuff stored inside here and a watertight area here. We see these in different type of vesicles and vacuoles and membrane layers. We see these in all types of cells. Another type of lipid is a steroid. You notice it because it has 4 fused rings. These are going to be carbon rings, and there are 4 fused. So you can see here each one has 4. I have different like off groups that come off, and that's going to be what changes my steroid. I want steroids that aren't going to dissolve in water. If I'm producing testosterone or estrogen, I don't want it to dissolve in water. I want it to be able to get where it needs to go. Cholesterol is going to be found in animal membranes. So what this does is really interesting. Cholesterol will actually get in between these hydrophobic heads and tails, and it's going to go in and it's going to allow it when the animal or the cell gets hot and naturally starts to expand. The cholesterol is going to hold it and pull everything together. And when the cell gets really cold and starts to kind of wants to freeze, the cholesterol is going to be inside and it's going to help push it apart. This also helps the membrane be fluid. It allows things to move and shift within this membrane due to this cholesterol. You notice it in membranes. So here you can see the cholesterol is down here in the hydrophobic area of the cell, but this is going to allow more fluidity and more flexibility inside of this membrane. Okay, that's it for lipids. Next ones are going to be proteins. Talk to you guys later. Bye.